to start with, give you a little bit of background for those of, we've all heard the name Robert ELSTON, but I'll give you a little bit of a background. He received his bachelor's and master's degrees in natural sciences from Cambridge University in England, then moved on to do a Ph.D. in animal breeding from Cornell University. From 1960 to the late 70s or so, he was at UNC Chapel Hill in biostatistics, and then he moved to New Orleans as professor and head of the department of what was known as biometry and genetics. In '95, he moved to his present location at Case Western Reserve University, where he and I were colleagues for a number of years. He has been the director of the department there in epidemiology and biostatistics for the last few years, and he's been the director of the division of genetic and molecular epidemiology for a number of years since he actually came to Case. He has a long and distinguished career, over 600 peer-reviewed papers, covering areas like segregation and linkage analysis, association analysis, GWAS studies, and other topics from a variety of different areas. He's been highly awarded throughout his career. He's received the Hoke Award from the American Psychopathological Association, the Wick Williams Award from the Fox Chase Cancer Center, the Leadership Award from the International Genetic Epidemiology Society, the William Allen Award from the American Society for Human Genetics, the Marvin Zellen Award from the School of Public Health at Harvard University. He's a fellow of the American Statistical Association, the Institute of Mathematical Statistics. He's a Guggenheim Fellow, a fellow of the Ohio Academy of Sciences. And the most impressive thing, I think, of among all of this is he's produced over 80 students and postdocs during his career that he's trained, as well as countless junior faculty that he's helped along the way, including myself. So with that, I'm going to pass it to Professor Vance to say a few more words.
Okay, I'm the color commentator for those of you watching the game tonight. Robert was my postdoctoral mentor, and I see some of my students out there, and you think it's bad meeting with me? You met once a month with Robert, and if you weren't prepared, you never came back. But I want to put in perspective all these awards and everything you won. I really see Robert as the father of modern statistical genetics. He took what was a cottage industry and through the ELSTON and Stewart algorithm made it a real discipline that has spawned all the things we're doing now, large GWAS studies, whole exome sequencing. This all started through his ELSTON and Stewart algorithm paper. Another interesting thing he taught me is, I guess when he submitted that paper, it was published in Human Heredity. And when he submitted it, he told us when we were students, I don't know if it's true, he goes, well, that journal didn't used to be a peer-reviewed journal. And he didn't even know it. And he sent ELSTON and Stewart in, and it still became a household word. So everybody who's worried, like Dr. Nye of publishing in Nature Genetics and Nature, et cetera, if it's good, and he always told us, if it's good, it'll get out there. Anyway, so it's just a real honor today for the students, the faculty, and everybody at University of Miami here to let you hear Robert and get some of his wisdom. It's a real pleasure for me to have him here, and even Jeff, who was a molecular biologist, sat through his statistics course and remembers it well. But so without further ado, thank you, Robert, and we're looking forward to your talk.
Thank you for those kind words of welcome. It is true that you publish a paper, it doesn't matter where you publish, eventually it gets the recognition it deserves. Even if it's in JAMA, all these things, it's not useless, it'll get that recognition of being completely forgotten. All right. So, and I know this is a very heterogeneous audience, so there'll be bits for everybody, and bits that'll confuse everybody, but hopefully we'll make it clear enough. It's really very simple if you just look at things simply. All right, so let's see. Introduction. So a genome-wide association study with case-controlled data. What we try and do is localize the disease susceptibility regions in the genome, okay? We got 3.2 billion spots, and we're trying to say it's somewhere in there. That's all it's trying to do. So these single nucleotide polymorphisms, they're markers there, those of the 3.2 billion that are variable, polymorphic, they've changed. And they usually have 2 alleles in them, which means there are 3 genotypes. We'll see that in a minute more clearly. And they've been used to cover the whole genome. So it's very simple. The idea is we look at everything, and then we want to say we've got a case control study. Can we say whether there's some areas of the genome that influence the particular disease that we're studying? So two kinds of tests have been applied. There's what is still done a lot, single marker association tests. And what that does is that basically looks at each one separately, one at a time, each of these markers, all right? And there's the multi-marker associated test, we look associated with infection status and multiple of these SNPs, these markers at a time. And the first, I suppose the first genome-wide association study about 4 years ago set a precedent. You look at them one marker at a time, and you use a certain model, and and everybody's done it the same since, and they say this is how you start. I've never ever understood that. It makes very little sense to me. So we got this out, and as you'll see towards the end, it's taken some time, but I've actually used it. And there's a lot to be said for not starting with one marker at a time. So typically, nowadays, you will have anywhere from, 6,000 to 1 million to 2 million markers, but spread out over the 3.2 billion things. All right. So, what kind of information is available? And one of my students drew this wonderful Venn diagram. I told him there's three things you can look at. You can look at the allele frequency difference. So it's called the allele frequency 10 test between cases and controls. All right. Another thing you can look at is there a difference in Hardy-Weinberg disequilibrium? And we'll see later on, the geneticists understand this Hardy-Weinberg disequilibrium, but we'll see that whereas the allele frequency is a linear, statistically, this other one just turns out to be quadratic, right? Wonderful. So the statisticians understand quadratic and the geneticists understand Hardy-Weinberg disequilibrium. Identical in this situation. And then you can look at the contrast in what is known as linkage disequilibrium between your cases and controls. For the statisticians, all you're contrasting is the correlations between cases and controls. Same thing, all right? And then you can do some together. So if you are here, I'm supposed to be close to this. If you are here, which this is saying you are testing for the difference in the allele frequencies, and also, that's the linear bit, and also the dominant bit. So now you have basically what's known as the genotypic frequency test, and that will be on 2 degrees of freedom because you're testing two things together. You can also have what is known as the haptotype-based test, assuming that there's no correlations. Hardy-Weinberg equilibrium. All right? So that's here. You can also, at least theoretically, you could look here. No one's ever done so. No one's given it a name. Probably good because it's probably pretty useless. All right? But conceptually, it's there, as you could do it. And lastly, you can do all three together, which corresponds to the phase-known genotype-based test. And that's, we'll see later, how that can be expressed statistically. All right? But those are all the possibilities. You look at each one at a time, two at a time, or all three together. All right. So these three tests, the allele frequency, Hardy-Weinberg disequilibrium, and linkage disequilibrium contrast test, are typically developed in what has been termed a retrospective context. This is the way the epidemiologists talk. In other words, the case control status is considered fixed, which is actually it was, and the genotypes are considered random, which is the way it was. But, you see, It's called retrospective because we believe it's not the cases and controls that determine these markers, but the markers control the other way around. Right? And the other way around we've always been able to do in statistics. For case-controlled data, we typically take advantage of the properties of the odds ratio, which means it doesn't really matter which margin we fix. When you've got two things, you get the same answer, and use the prospective logistic regression model. Make the case control status the random variable dependent on the predictors, the markers. All right? So this, and it's very interesting. Typically, well, this sometimes is done now, I suppose. I think a paper may come out saying this is the only thing you should do. Not by me, but I just declined to review this paper because I just don't have time. But I saw it by a good friend of mine. He's written it. And it's probably going to say, why are you doing what everybody else is doing? That's what I've been saying all along. Why? I don't understand people who just do it because it's so-called state-of-the-art. To me, that's not good enough. All right? State-of-the-art. What you want is something that answers the question you're asking, not something that everybody else does. So that's my message to you. Don't do what everybody else does because that is state-of-the-art. Chances are, in a few years' time, it won't be, and you'll be criticized for it. All right. So the idea of prospective modeling, it allows for much greater flexibility, especially if you want to put covariance in the model, in logistic regression, and it provides a natural way to adjust for any correlations between the tests or other covariates, which we shall see. All right. So we need some notation and assumptions. And for those of you, maybe there's still one or two who are not too happy with all this algebra and stuff, anything that's down here algebraically is meaningless if I can't put it into words. All right? And when I read some of these papers, they don't tell me what their symbols are, I just give up. I give up completely because I don't know what they're talking about. And I read all these papers. So I'm going to tell you what these things mean. All right? So we're going to have two diallelic SNP markers. Okay? We'll see, for those of you who don't know the word diallelic of SNP, we'll just say that there are two of these things called alleles, and each person has two of them. Two A's, we'll call them A1 and A2, and somewhere else you have two B's, B1 and B2, somewhere in the genome. Okay? And the A1, because if you have two, all right, one is more frequent than the other, clearly. You think of the proportions are, you know, they've got to add up to one, and the minor allele is considered the one with the smaller frequency. All right. Oh, geneticists use the word frequency when they mean relative frequency. Everybody knows a frequency is a count. Geneticists have never learned that. I am very fussy about getting your words right. All right? Geneticists don't care, even statisticians don't care, and epidemiologists don't care, which means that nobody can understand what the other person's talking about. It's a wonderful Tower of Babel we have. All right. And if anything is not clear as we go through, raise your hand. All right? I've told you some things you don't have to understand, depending on whether you're a statistician or geneticist, but I think it'll become clearer. So we have a random variable x. If the person is A1, that has a value of 1. Person has A1, A2, one of each of these, it's a 0. And for -1 for A2, A2.